these are the paste of banana and onion. Now students, before starting the experiment, we must aware about what is DNA. We all know that DNA is the genetic material. And if we talk about the full form of DNA, then it is deoxyribonucleic acid. And we know that all cells of plants, animals, and all living organisms consist of DNA. So here we are going to isolate DNA from plant sources. Now students, let us study what are the different materials required for DNA isolation. So here you can see that number one is plant sources. So we can take banana, we can take papaya, we can take onion, we can take strawberry. And strawberries are the best because they have not much of DNAs, not much of chromosomes. Now then we need table salt or sodium acetate. Here I have sodium acetate. Now then we need beakers. So here you can see beakers are required. Here you can see beakers are required. Now here we need coffee stainers. You can see the coffee stainers. You can make coffee stainers at home by the help of paper napkin. Now another one is ethanol. Here we need ethanol. Here you can see ethanol. And here we need chill ethanol. Why? I'll let you know. Glass rod is required. So here we need glass rod for spooling. What is spooling? That I also going to explain today. Now centrifuge machine. Here we can see mini centrifuge machine. This is the centrifuge machine just for centrifugal force, applying centrifugal force to the molecules. Now students, here let us start the experiment. Now what are the steps required? Here you can see some enzymes are required on the top. Because you are performing this experiment at home, so you can take some alternatives of enzymes. In place of lipases, you can take shampoo or Vim liquid. Here you can see the Vim liquid. Okay? Or any liquid soap you can take because it consists of lipase enzyme or those chemicals which can remove lipids or which can digest lipids or oils. Now then we need island cleaner. Here I have island cleaner. Or you can take pineapple juice, papaya or meat tenderizer. Why? Because these molecules or these chemicals can digest protein. So any one you can take which is available at home. So here I have taken island cleaner. Got it? Now students come to the experiment. Now here you can see what are the steps. So this is a technical way of isolating the DNA. Here we are going to isolate DNA at home. How we can isolate DNA at home? By simple chemicals available at home. So here you can see students, see this is the paste of banana. I'm going to filter it. Here you can see I'm putting it in the filtrate. Here I'm putting this in the coffee stainer and you can see how the filtrate comes out. And the filtrate has our DNA. Now, another thing here you can see, this is already filtered. This is the onion paste which is already filtered out through the measuring cylinder. Now students, what is required? Next is required is you just pour this filtrate in a beaker. So let us say this onion filtrate we are going to pour in a beaker. Now, after pouring it in beaker, you add sodium citrate, half tablespoon sodium citrate. So what is the role of sodium citrate? The role of sodium citrate is it adjusts cell wall. Along with that, you use Vim liquid. Vim liquid has lipase-like activity, chemicals which have lipase-like activity. So here lipids get dissolved. We know that DNA is covered by plasma membrane which is made up of lipids as well as it is present inside the nucleus. In eukaryotic cells, because plant cells are eukaryotic cells, so DNA DNA is wrapped within a nuclear membrane. It is present within the nucleus, which is surrounded by nuclear membrane, and nuclear membrane also has lipids. So here we dissolve the lipids. Now to dissolve protein content, we can use papain enzyme, which is present in papaya, or we can use pineapple enzyme, which also has protein digesting enzyme, or we can use iron screener or meat tenderizer, which also going to digest proteins. So here I am using 4 to 5 drops of iron screener and then stir it well. Stirring should be done very gently, just to avoid breakage of DNA fibers. Now banana filtrate is already chemically treated. Here you can see this is already chemically treated. Now students, these are microtubes. Here you can see these are microtubes. Now for some content, small content you can see in the microtubes. Here you can see small content of this filtrate, this banana filtrate in the microtubes, and place it in the centrifuge machine. Same with this, take small quantity of onion paste, onion filtrate, chemically treated onion filtrate, and this is also to be placed in centrifuge machine. Then you are going to place it, cover it, and then on the switch. Now for 1 minute you just allow to centrifuge the content. What is the role of centrifugation? Centrifugation separates the molecule on the basis of centrifugal force, and after centrifugation, when we allow to settle down the molecules, they settle down as per their molecular weight. And students, as we know that we have digested all the macromolecules like proteins, like lipids, we digested all the macromolecules. So what is left with us is DNA, which is macromolecule left undigested, because we are going to isolate DNA. So this molecule is left undigested, and after centrifugation, molecules settle down as per their molecular weight. So here you can see that only what is left over here at the bottom is DNA. Now students, after 1 minute, you can take out the microtubes. Here I am going to take out the microtubes. And to these microtubes, so here we have gel-ethanol. So add it on the side walls of the tube. And there you can see DNA is going to precipitate out. Here you can see if it takes time little bit. Here you can see this is the DNA. This is DNA of banana. Here you can see this is DNA of banana, and I'm going to wrap it on a rod on a glass tube. This is DNA of banana. So here you can see, this is what is DNA of banana. Now this is onion paste. Now take out. Here students, you can see the threads of DNA. And the last step, here you can see. Here you can see the threads of DNA. Here you can see the threads of DNA. These are the threads of DNA. And when I wrap DNA threads on a glass dot, then this process is called spooling. So this is, you can isolate DNA from different plant sources. So what to write in your practical file, let us study about that. So students, here you can see what are the steps required for isolation of DNA. So here you can see, first of all, you take small amount of plant material, grind it with the help of pestle and mortar, or you can make a paste in mixie or by the help of airtight or you can say polybags. Now break the cell wall and other alveoli of the plant cell. Here some enzymes are mentioned. So just to avoid enzymes, here you can see some alternative chemicals are required in place of enzyme. Like you can use liquid detergents, shampoo you can use iron cleaner or pineapple juice or papaya or meat tenderizer, which is used to soften the meat. So in this way we can use different chemicals in place of enzymes, because enzymes are very costly as well as they are not available at home. So just to perform this experiment at home, you can use different alternatives of enzymes. Now students, finally we can see how spooling is done. DNA is pulled out around the glass rod, and then what are the precautions? You are supposed to take all the materials and chemicals in appropriate amount. Stir the content very gently, else if you forcefully stir the content, then the DNA fragments may be broken down. And chill ethanol is used. If you use warm ethanol, then, or you can say normal temperature ethanol, then it takes time to appear the DNA, or because in alcohol DNA is not soluble, it makes precipitate of DNA. So this is what about your practical. Now there are some questions which you are supposed to go through. For example, what is DNA? So here you are supposed to expand DNA. Expand means you are supposed to write the full form of DNA. So all these questions we are going to discuss on Saturday in practical online period. So here you can see, name the enzymes used to remove DNA from plant sources, name the enzyme which digests DNA molecule, and is this the enzyme used in this process? No, DNA digestive enzyme, which is DNase, is not required to remove DNA. Then what is the role of chill ethanol in this technique? Because DNA is soluble in water, so here chill ethanol is going to precipitate DNA, because in you can say chill ethanol, DNA gets precipitated out. And spooling means on the glass rod you can wrap DNA molecule. So this is what about the practical. Thank you. Have a nice day.